Hi everyone, my name is Elaine Martis and I'm really pleased to be asked to present this keynote lecture for the Cancer Genomics Consortium meeting in 2020. The title of my talk today is Cancer Genomics Drives Pediatric Cancer Precision Medicine. The following disclosures for your information. I want to set the stage for talking about pediatric cancer precision medicine by reviewing just briefly the situation in adult cancers, where genomics really provides a treatment playbook for many of the very common cancers seen in the adult setting. A great example of this shown here is non-small cell lung cancer, and really the pie graphs on the left-hand side of the slide illustrate the transition that we've had over the course of large-scale cancer discovery from where we started, which was really sort of gross pathology-based descriptions of lung cancers based on histology, to now where we can take the majority of lung cancers, adenocarcinomas, and really break them down according to the different genetic drivers that have been identified, and for which in the same time frame really a lot of targeted therapies have been developed to address these cancer drivers. And what this really has resulted in is the current situation in terms of diagnosis of a patient initially by imaging, and in a secondary sense using some sort of a biopsy procedure illustrated here, which is followed by what we could call genome analysis. This takes place in the context of anything from a hotspot mutation panel all the way to whole genome and RNA sequencing. But in this setting, what the analysis may reveal is, for example, the presence of an EGFR driver mutation, in which case the patient would be treated with a receptor tyrosine kinase inhibitor, of which there are many now available and FDA approved. In a setting where both EGFR and KRAS mutations were identified, in the current day, this patient would receive immune therapy, typically with or without a chemotherapy combination. And then we now know that there are several fusion drivers in lung cancer, such as ALK or ROS fusions. And in this particular case, there are also correspondingly many targeted therapies available for these specific fusions. And so the bottom line is that genome analysis is really playing a significant role in the treatment decision-making paradigm for lung cancer. Just as an example, in adult cancers. If you look more broadly in this tumor to normal comparison using next generation sequencing, as Mike Berger and I wrote about in Nature Reviews Clinical Oncology a couple of years ago, really the determination of somatic and germline variants shown here now can dictate a number of different possibilities in the therapeutic paradigm, which are illustrated for both somatic and germline, as I mentioned, where really the newest developments here are in the realm of immunotherapies and high mutation load tumors, in some cases NeoAntigen Prediction for vaccine development, and really even the germline is now telling us much more about susceptibility, and in some cases really is indicative again of therapeutic interventions, as illustrated in this figure. This is now in stark contrast to the pediatric setting, where we have pediatric cancers typically having a very low number of mutational lesions, and these typically yield few, if any, druggable targets from just DNA profiling alone. In addition, treatment with FDA-approved drugs is difficult in pediatric patients. We have issues with dosage and unknown adverse event profiles, for example, where it's often tricky to find the right dose for the patients, even if it's indicated genetically. We also know that, at least in the past, clinical trial enrollment has excluded pediatric patients, which is changing a bit with new legislation that's coming online this year in 2020, but I think there will still be some limitations in place. And finally, in this new realm of immunotherapeutics that I referred to earlier, really checkpoint blockade therapies are typically not considered applicable in the pediatric setting, although I'll talk about an exception to that at the end of the talk today. Although there are CAR T therapies that are approved for leukemias and lymphomas. So there is some action in the immunotherapy space, and I'll talk also about some preclinical work that we're doing to develop new pediatric immunotherapies a little bit later on in the talk. So pediatric cancer remains a very challenging problem for us. Cancer is the leading cause of death by disease in the developed world in children over 1 year of age. And the work I'll talk about today is really meant to try to usher in an era of increasingly looking for molecular targets in pediatric cancer, much like I described in adults. I'm using a variety of different assays, which I'll talk through in the context of some of these challenges that I'll already mention and which I'll talk about a little bit more in detail. In particular, we want to move the needle on the use of targeted therapies and immunotherapies for pediatric cancers, because even in cancer types such as acute lymphoblastic leukemia, where children have a very high cure rate, they typically experience significant health problems later in life because of the aggressiveness of chemotherapy that's used. So we have a long history in pediatric cancer genome discovery through the Pediatric Cancer Genome Project, done over a 5-year period with St. Jude Pediatric Cancer Hospital. And what's shown here is sort of a summary article in 2012 at the end of that project, and illustrates some of the characteristics that were identified. As I mentioned previously, pediatric cancers do have very few point mutations. On the other hand, they have many tumor types which have common fusion gene drivers. I'll talk a little bit more about these later. In particular, it's also the case that pediatric cancers, because of the low mutation load, actually are very epigenetically driven cancers. This includes alterations to histone proteins that change chromatin packaging, or other epigenetic drivers such as differential methylation that really result in modified transcriptional programs that drive cancer onset and progression. And because of these two facts, fusion drivers and transcriptional program modification, as I'll talk about a lot today, studying RNA from pediatric cancers is really becoming a significant driver in terms of helping us to identify ways to understand and characterize and treat these patients. Finally, we talk a lot about germline contribution in the adult setting, but germline contribution in pediatric setting is actually more significant than previously thought. Upwards of 10 to 12% of pediatric cancers, possibly more once we learn more of the susceptibility genes that have yet to be discovered, are driven by germline predisposition. This is another article, not our work, but a paper from Nature a couple of years ago, really comparing now post large-scale discovery in adults and pediatric cancers, the significantly mutated genes, or SMGs. Again, from this plot series on the left-hand side of the slide, you see the difference between the number of significantly mutated genes per sample. It's a little redundant. And in the pediatrics versus adults. And then you also see by the Venn diagram on the right-hand side that the overlap in significantly mutated genes is actually quite small. And many of these in the gray area are either undruggable targets such as KRAS, at least at this point in time, although there are new drugs coming online, or they're actually cancer tumor suppressors. And so in aggregate, this gives us not much to work from in many cases. So in response to these challenges that I've just laid out, a couple of years ago, after joining the faculty at Nationwide Children's Hospital, we put together a protocol called the IGM Cancer Protocol, which is shown in outline here in terms of the various steps that are utilized. The first thing that we put together to really record all of the information that's seen in this workflow was a REDCap database that underlies all of the activities that are shown here and is used to really record all the steps in the process, what their status is at any given point in time, and then to warehouse the data that's being produced through this approach. You'll see on the far left-hand side of the slide that the first step in the process is really getting one of our cancer providers to complete a simple one-page nomination form. This is a small number of query boxes, radio buttons that need to be filled in, and the completion of the form then triggers the study team to be notified that there is a completed nomination. We then evaluate that nomination and vote whether to accept the patient onto the protocol or to decline. And there are a variety of reasons for declining, but typically these patients are accepted, and then the family is approached for consent by our genetic counselor. Once consented, we then work with pathology as well as other providers in the hospital to obtain samples from the patient, tumor and normal, and also from the family, in particular from mom and dad. We do try to obtain either a blood or saliva sample, which we won't study directly through the early parts of the protocol, but in the case of an identified germline susceptibility, we would go back to those samples if appropriately consented to evaluate the heritability. Once we extract nucleic acids from these samples, DNA and RNA from the tumor and DNA from the normal, we then proceed with sequencing data analysis. And in any case where we find a medically meaningful finding, which I'll talk about, we can confirm this in our CAP-CLIA lab and then write that information into the electronic medical record. Our REDCap database also connects to the EPIC EMR so that we can get pathology reports and other information from prior testing if available, and also allows us to connect into the radiology image database to look at scans over time for these patients if we're putting together a case series. Finally, we do house de-identified information and analysis results on our Confluence site, which allows many individuals to produce analyses and paste these into the Confluence site. And this is a good quick reference for these patient cases. The genomic profiling that takes place, I've already talked about a little bit. DNA and RNA sequencing, data analysis, and variant annotation and interpretation and reporting are the final steps shown in the stepwise flow diagram at the top. The eligibility onto this protocol is primarily patients with rare cancers, relapse or treatment refractory cancers, and in some cases rare hematologic diseases have been studied as well. And the assays that we perform are outlined here. First, we have what I call enhanced tumor normal exome. What this means is that in addition to the exome capture probes, we add in copy number probes that tile across all the chromosomes, providing about 30 kb resolution. And this allows us to understand chromosome arm level gains or losses, as well as focal gains and losses. We try to accumulate about 250-fold coverage depth on the tumor and normal DNA so that we have good sensitivity. And of course, we can use these data to call germline and somatic alterations. RNA-seq I mentioned earlier. We studied the RNA data from the tumor, and we perform multiple analyses on these. I'll walk you through those. And then finally, we do have a quick turnaround test from ArcherDX that allows us either in the heme malignancies or in solid tissue malignancies to identify driver fusions in a rapid clinical test that has about a 1-week turnaround time. How do we analyze all these data? Well, we have set up Docker containers in the Amazon Web Services cloud with these computational analysis modules. At the top, you see the enhanced exome sequencing of tumor normal going through our Churchill aligner for both germline and somatic identification of point mutations, indels, structural variants, copy number variants, as well as loss of heterozygosity through multiple analytical pipelines. And then on the bottom, the tumor RNA-seq also goes through Churchill aligner. We also look at differential expression. We use Salmon to convert these gene expression values to TPM and analyze each individual patient compared to a large database of patients at the Treehouse consortium to really firm up the diagnosis and better understand where this patient's RNA-seq expression groups them. And again, this is from the standpoint of having a lot of epigenetic drivers in the mix, but really the readout in the cancer biology being in the RNA data itself. And so from the RNA-seq data, we also can detect fusions. I'll talk a little bit about that because they're so important. And of course, this would be compared to the Archer results that we get from this patient. And then finally, another interest in terms of my own research is looking at the tumor microenvironment. We can do this approach using the RNA-seq data and RNA-seq-based deconvolution, which I'll also mention here. Fusion detection. First of all, I've already emphasized the importance of fusions in the pediatric setting. So we want to be able to detect these with a high quality, low false positive, low false negative fusion detection pipeline, if you will. And of course, as shown here, there are a variety of different fusion detection approaches, but they all have sort of strengths and weaknesses. And so I think this is a difficult problem, especially in the clinical setting. And so what we've decided to do in an approach that will be going forward for publication soon is to really combine the best-in-class fusion detection algorithms shown here across 6 different algorithms, using the same RNA sequencing data set, of course, and then use an Ensemble script to sort of evaluate the overlaps in called fusions to identify the commonalities between the different fusion detection algorithms. And if we can essentially detect a fusion with 3 or more of these algorithms, we typically have a strong candidate. These data then go through a strategy including prioritization, comparison of the fusions to a knowledge base, which tells us about known sources of false positivity and allows us to subtract out fusions that we know are false positives, but also to evaluate read support and compare these fusions to a database or whitelist of known fusions and fusion partner genes, so that even in the setting of having low read support, something that looks like a real fusion can get elevated back into the output and be evaluated manually. And so this whitelist comparison rescues known fusions that fall below the parameters of read support. And at the end of the process, if we find something that's medically meaningful, we can again put this through a CAP-CLIA validation of directed RT-PCR and Sanger to validate the fusion in our CAP-CLIA lab. The results of this pipeline for 181 patients in our cohort are shown here. This is indicative of the number of cases where a particular fusion has been found, and the black bars indicate known fusions, whereas the red bars indicate novel fusions that have been identified. And these fusions, as I mentioned, have been clinically confirmed as well. So this has been, I think, a very valuable part of our data analysis pipeline for the RNA-seq data. Another way that we evaluate RNA-seq is using the tools that are and databases that are available at the Treehouse initiative at UC Santa Cruz, shown here with their URL. And this is a very nice compilation of a variety of important tools, including the tumor map, which is the principal components analysis that I mentioned earlier that we utilize on a pretty frequent basis, and a housing of data from RNA-seq from tumor samples across both adult and pediatric cancers. And we actively contribute data from our pediatric cancer RNA-seq into this database as well, often to enhance the data fields around very rare cancers in particular. So I'll give you some examples of how we use the Treehouse database in the following slides. One way that we evaluate our individual patients is by gene expression outlier analysis, and of course these evaluations require comparisons back to other cancers because we often or never have the luxury of having adjacent non-malignant tissue from which to extract RNA and do those direct patient-specific comparisons. So here what we're doing is we're essentially taking our RNA-seq data, we analyze it using Salmon to obtain transcripts per million values, and we can either evaluate versus the Treehouse cohort or a subset thereof, for example, all CNS tumors, all Ewing sarcomas, or we can just evaluate back to similarly diagnosed patients within our IgM patient cohort. The way that we do this is after running salmon, we will just filter out data for the protein coding genes, do some quantile normalization, and calculate differential expression per patient using the formula that's shown below on this slide. And then we can work it through a variety of different analyses to identify druggable gene targets based on outlier expression using the drug-gene interaction database, or take the data through ingenuity pathway analysis. And I'll show you an example of both of these. In particular, the data that I was showing here is actually data that is analyzed versus the Treehouse. This is from a patient with Ewing sarcoma, and you can see by this box and whiskers plot that this patient expression of JAK1 maps nicely to other Ewing sarcomas. The red dot is the patient under study. And then by comparing to the Treehouse cohort here, or sorry, to our own internal cohort here, you can see that JAK1 is commonly expressed in other heme malignancies, but in particular for non-CNS solid tissues such as the Ewing sarcoma, this particular patient has a very high expression level of JAK1 compared to the other patients in our cohort. So this is just one example of where we come by potentially druggable gene expression outliers. Often these can go forward with an immunohistochemistry confirmation of overexpression, in which case the targeted therapy would be indicated. Finally, in a non-sequencing, non-NGS-based mode, I should say, we also have started to perform classifier-based subtyping using methylation array data and the CNS classifier, that's a machine learning classifier available from the DKFC in Heidelberg and recently published in Nature. And this classifier provides us with subtyping refinement or validates pathology-based diagnoses, and can also generate copy number plots as well as information about MGMT promoter methylation status, all of which are important in the context of CNS tumors. Another evaluation of RNA then goes to identifying changes in the tumor microenvironment, the presence of immune cell infiltration in particular. Here we utilize a combination of CyberSort, which is shown here from the two publications in 2015 and 2019, as well as Timer, which comes from Shirley Liu's lab, to really shape out the deconvolution of RNA-seq data and help to identify by permutation analysis and matching to known expression profiles of different immune cell types, which immune cells are present. And I'll show you an example of this later in the context of longitudinal samples comparing the changes in immune infiltration in a primary compared to a recurrent tumor. Finally, from our own work with the laboratory of Obi and Malachi Griffith, and in a collaboration with Bob Schreiber, all at Washington University, we've developed these pVACtools approaches that were published recently in Cancer Immunology Research. This is a set of tools really that is centered around neoantigen prediction in many forms and flavors, including the original pVAC-seq algorithm that we published in 2016, but now expanding out to a variety of different tools that interconnect using an API, including a browser-based GUI that called pVACviz that actually provides very intuitive interface to neoantigen prediction without need for extensive bioinformatics training. And in many ways, in our pediatric cancer cohort, because of the presence of fusion drivers, we've been using pVACfuse to evaluate pediatric fusion junction sequences. Here's just some data shown here generated by my postdoc, Katie Miller, where we look at a variety of different cancer diagnoses, the corresponding gene fusion that's been identified, and using the junction peptide between these two in-frame fusion partners in every case, we've been able to predict high affinity neoantigens where we see the combination of gene 1 and gene 2 amino acid sequences at that junction point. And so this is fuel for future studies in immunotherapies that I'll talk about just in a little bit. Finally, I just want to finish up this part of the talk by saying that the protocol enrollment has been rapid and increasing over time, as you can see from this graphic, since we opened the protocol in January of 2018. And over the next year and a half or so, we plan to make portions of this protocol, including the tumor normal exome that I talked about, and ultimately RNA sequencing, as well as the methylation profiling, part of our CAP-CLIA lab, so that these turn from research-based testing into clinical testing. To do that, we really want to drive the reasons behind the clinical validity and clinical value of these tests. And we do that by evaluating the impact of the cancer protocol on the patients that we've studied so far. So here's a recent compilation of where we're at in terms of clinical impact in the realm of providing diagnostic, prognostic, or therapeutically relevant findings for the patients that have been studied so far. So this shows a summary for the first 152 patients that have been sequenced and analyzed completely through clinical confirmation, where we can see in the gold boxes at the bottom that we can identify a variety of different types of findings, including germline predisposition, somatic fusions, which I've talked a lot about, potentially targetable expression, which I mentioned a little bit ago, medically meaningful copy number variation, these chromosome arm level gains or losses that can be prognostic and help determine this, how aggressive the treatment is the patient receives. And then finally, medically meaningful somatic variation is often identified as well. And so we feel like the impact of this has been significant, and in aggregate, given the numbers that I'm showing here, we can, for 93% of the patients studied to date, identify at least one medically meaningful finding from the study of those patients. So I want to give you an idea now, going through some case studies, how all of these pieces tie together in the practice of precision cancer medicine for pediatric patients. So I'll start this with our patient shown here, CH21, a child that was diagnosed in 2013 at age 1 year of age with this rare meningeal sarcoma, which you can see on the leftmost MRI. It was removed surgically by gross total resection, shown in the post-op MRI, and the patient went on to receive some proton therapy as a follow-on to the surgery. No chemotherapy was utilized. The patient sample went for DNA panel testing at a commercial entity, and there were 2 results that were returned in the child's report. One had identified a pathogenic mutation in an intestinal cell kinase, ICK. The second was a variant of unknown significance in platelet-derived growth factor beta. So there was nothing really to be done with this information at the time, and of course the child needed to fail the standard of care to utilize further genetic profiling at that point in time, and so proton therapy was used, as I mentioned, and there was periodic imaging that occurred. That imaging in 2018, when the child was 5 years old, showed a recurrence in the same area of the same type of cancer, meningeal sarcoma, as I mentioned. And again, this was removed by gross total resection, and at that point in time, the patient was evaluated through the IGM cancer protocol, as I've described it for you already. We were able to identify these same 2 mutations in the context of the commercial testing that I mentioned earlier, but because the commercial testing only evaluated the tumor DNA, not the normal, as you can see on the left-hand IGV profile, the intestinal cell kinase mutation is a deletion of 12 bases that's actually a germline based, potentially predisposition alteration, although it has not been reported extensively in cancer. In contrast, the platelet-derived growth factor beta is a somatic in-frame deletion that is not a variant of unknown significance. It's been seen multiple occasions, and it's a very interesting deletion in that it actually occurs in this juxtamembrane domain, which is shown on the far right-hand side in a profile of what the protein structure for platelet-derived growth factor beta looks like. This is in an autoinhibitory domain that in the normal unaltered protein masks the catalytic domain, but in this alteration actually continuously fires the protein kinase activity. And so this is a predicted gain-of-function mutation that is the driver for this patient's cancer. We looked also in the expressed RNA for PDGFRB, and we're able to show in the dark blue bar here that this patient is among the highest expressors. The other two patients you see that are even higher also have similar alterations in this gene. And we also were able to show from ingenuity pathway analysis that the platelet-derived growth factor beta alteration lies at the heart of this very activated pathway, as illustrated by the dark orange and red symbols on the IPA diagram. So based on this information and clinical confirmation of the PDGFR beta alteration, we were able to take this information back to the child's oncologist using data such as shown here, indicative of membrane-bound RTKs like PDGFR beta, being the source of activated pathways. The oncologist was able to identify an intermediate, which is dasatinib, and this was provided to the child as an orally available therapy starting in September of 2018 following our information being provided back. And you can see that this patient, as of June of this year, continues to be healthy with no evidence of disease. Here's another case. Now I'm looking again in the CNS with an atypical neuroepithelial tumor. This is a longitudinal study, a primary and a recurrent cancer from the same patient. And what you can see here is the patient originally presented at age 10 in year 2015 with a mixed glioneuronal tumor. A recurrence in 2017 again was removed by surgery and called by pathology a high-grade neuroepithelial tumor. Also, the slides were sent to St. Jude, and they categorized this tumor type as anaplastic ganglioglioma. By the principal components analysis shown on the right-hand side, you can see that the two tumors, the primary and the recurrent, grouped reasonably close to one another, but admittedly in a fairly sparse area without a lot of samples to really provide much differentiation. So we studied the primary tumor and the recurrent tumor as well, and didn't really identify any mutations from the DNA-based analysis, but RNA analysis identified this unique EP300B core fusion that we were able to not only confirm clinically, but also show that was present both in the primary and the recurrent cancer. If we take a little closer look at this fusion, you can see that the lower diagram illustrates the place where EP300 and B core are fused on the bottom, labeled JL36. On the top, you can see a differential fusion that we were able to identify from a TCGA sample, an oligodendroglioma, that also had this EP300 B core fusion. So at the time that we identified this, this was really the only other example that we were able to find in the peer-reviewed literature for this novel fusion. We looked a little further in an effort to try and understand what this fusion might be doing or whether it was indeed pathogenic. The best we were able to do at the time was this 2016 paper that identified a series of 10 samples with a CNS high-grade neuroepithelial tumor that had a B-core internal tandem duplication, ITD. And based on this paper and some of the downstream manifestations for this B-core ITD, we were able to come up with a hypothesis that maybe this EP300 B-core translocation was actually biologically similar to the B-core internal tandem duplication. And we pursued that a little bit further, but didn't produce any medically meaningful information at the time just because this fusion appeared to be so rare. One of the downstream analyses that we were able to do, however, was to look at gene outlier expression, which I described earlier, and in this case, the candidate that came up was epidermal growth factor receptor. In this bar graph shown here, you can see the two samples from the patient. The primary had extraordinarily high EGFR expression, the recurrent less so, but definitely they were overexpressing EGFR compared to many of the samples at the time in our tumor cohort. And in the fullness of time, this patient did recur in their cancer, and we were able to then go back to the literature and identify this paper from early 2019 reporting a recurrent EP300B core fusion to the tune of 3 different patients. So our patient is now, at this time, the 4th patient who's been identified with a pediatric glioma in this particular alteration. But similarly to our findings in EGFR, this series of 3 patients also were indicative of elevated EGFR expression. Of course, this is a very targetable alteration, so we were able to confirm this by immunohistochemistry in the pathology laboratory, and this patient is receiving cetuximab at this time. Taking a little bit closer look at the tumor microenvironment now for this longitudinal samples set, you can see 3 lines of evidence shown here that there is increased immune infiltration in the recurrent tumor. On the left-hand side, we see analyses from Estimate, the algorithm that is often used to look at tumor purity, but also calculates an immune score. And you can see this is negative for the primary tumor on the top view and very positive for the immune score of the recurrent tumor. This is further reinforced in the center. Our CyberSort analysis shown here shows a significant p-value for the recurrent tumor labeled R with the infiltration of M2 macrophages as well as monocytes compared to the primary tumor labeled P. And then on the rightmost side, we have NanoString Immunoprofiler, which is an RNA-based analysis of immune cell types for the primary, again labeled by P, which is fairly immune cold, as illustrated by the blue bars, and for the recurrent, which is fairly immune hot, illustrated by the orange bars for a variety of different immune cell types. So we followed this up a little bit more by looking at the changes in neoantigen load. On the left-hand side, you can see that the variant allele frequency and the numbers of variants is quite different for the recurrent tumor on the y-axis compared to the primary tumor on the x-axis. And indeed, we took these mutations, both in the primary and the recurrent tumor, and compared them using pVACtools for MHC binding affinity, and show here that in the recurrent tumor, as illustrated by the red dots on the graph, we see a significant uptick in the number of strong binders from these additional mutations that have occurred in the course of the cancer's recurrence. So these types of observations, as well as the fusion neoantigens that I talked about earlier, and the ability to use RNA as a source of information about the immune microenvironment and changes therein, have really led, along with important work done by collaborators at our institution, to this recent award of NCI funding under the Cancer Moonshot. And this is really a preclinical grant to evaluate a variety of mouse models, both with CNS cancers as well as sarcomas, using a variety of different novel immunotherapy approaches. So engineered natural killer cells, oncolytic virus-based therapies, and also using drugs that target the myeloid cell-based immunosuppression that we are identifying, much like the patient that I just showed you. Our role in this will be to apply a variety of different tools, some of which I've just told you about, some of which I don't have time to talk about, to provide a comprehensive immunogenomic profile in the pre versus post-therapeutic setting for these animal models, and to hopefully progress some of these therapy approaches into clinical trials. So I'll just finish with a more recent case that we identified that is fairly rare in the pediatric setting and was quite interesting. And this is ultramutation in a brain tumor with Lynch association in a glioblastoma. Here's the case history, and pay attention to the dates on this one because this patient presented in the fall of 2019 with a high-grade neuroepithelial tumor, WHO grade 4, at 13 years of age. This was removed, and the postoperative MRI is shown here, and the follow-up MRI just a month later shows, again, significant tumor development in the exact same site as the prior tumor. The patient was treated with focal radiation and veliparib, followed by a course of temozolomide, but unfortunately, upon follow-up in January of this year, showed progression. And so the tumor was again operated on, and we received tissue, which we studied under our IgM cancer protocol. What we were showing from our germline analysis is here. The patient has an inherited PMS2 alteration, which is a pathogenic founder variant, and is associated with Lynch syndrome. As many of you know, these genes, MSH2, 6, MLH1, PMS2, and POLE, are associated with Lynch syndrome, and this is an increased risk because of mismatch repair defects for many different tumor types. In the somatic profiling, we were able to identify the corresponding PMS2 variant, the second hit, if you will, and this was then reflexed to immunohistochemistry-based evaluation of PMS2 staining that verified there was no PMS2 being produced in this tumor. We also identified a POLE and several MSH gene alterations as well at varying variant allele fractions. But ultimately, this is an ultra-mutated glioblastoma, so over 14,000 high-quality variants were detected in the exome data that we produced. Again, you can see some known cancer genes with high and low VAF mutations that were identified, but this is really the detritus of mismatch repair defects from the PMS2 alterations that I talked about already. We also profiled this patient with the methylation profiler and came back with a very strong score indicative of glioblastoma IDH wild type subclass midline, as illustrated by the high confidence score as well as by the Tisney clustering here with a group of other IDH wild type glioblastomas. So the diagnosis on this patient is ultramutated glioblastoma. And you can see that, of course, this invokes the use of immunotherapy, so the patient is currently taking nivolumab and being watched very carefully. As well, because of the PMS2 germline alteration, genetic counseling was in place for the family, and they're being tested as well, and there's heightened surveillance for this proband as he goes forward on his treatment. So just to conclude then, I've tried to show that the clinical genomic assays of pediatric cancers can really add important clinical evidence, and in many cases where we're studying, these are impacting treatment planning, diagnosis, and prognosis. We've combined DNA and RNA data to provide powerful information about cancer drivers, about altered gene expression, and also in the tumor microenvironment, which has led to new research. And given the rarity of these pediatric cancers, we really are calling for renewed efforts to share data, much like the Treehouse Consortium. We're also depositing all of our data at dbGaP. And we're trying to champion the provision of internet-based analytical and decision support tools, such as the Docker containers that we use at our Amazon Web Services, such as the nice tools that are provided for methylation classification by the DKFZ, and such as the Treehouse cohort that we talked about at the UC Santa Cruz site. So I think these are all important mechanisms to help even the playing field and ensure that kids everywhere can get the best care possible for their cancer.